All right, so today we're going to talk about the components of the central nervous system and hopefully the components of the peripheral nervous system as well. And then on Wednesday, we'll move on to talk about a little bit more peripheral nervous system and senses. So we're going to be working our way through from the brain on down and focusing on overall functions of each structure. So we'll start with the brain and how we divide up the brain and how those divisions correspond to the functions of the different regions, as well as all of the protections that keep the brain as protected as possible. And when I was finding images for you guys, I did find this website, which has a lot of nice little overviews. So if all of this is sounding like too much, maybe start with some of the information on here to help ease you in. So there's some things that I hope get cleared up. Either you may have noticed in your readings already or that we touch on today. There's a lot of myths regarding the human body and the human brain, and these are just a couple of them that I feel like come up more. So this is certainly not inclusive, but for one thing, there's a big one about how like we only use a small percentage of our brain power, and then that gets used and in like movie plots to say, well, what if we sped that up? But it's not true. We really do use all of our brains most of the time, if not all of the time. They all have their own functions and purposes, and we wouldn't quite be the same if we didn't have all of them. Another one that we probably won't quite get to because it's too much detail, but we feel like when we remember things that it's accurate, that memories are actually like video of our past. But it turns out that the way memories work, that's not true. When we remember things, we are sort of remaking the experience every single time we recall it, and it's pretty easy to change your memories over time, especially in light of new experiences and new knowledge. So we know that memory is not very accurate, and that has pretty profound implications as far as what happens when we ask people to recall things in different circumstances. Another one is that our senses tell us exactly how the world is. That like really we're just these walking video cameras, right? And we've got some video recordings in here, and we've got good, microphones in there and all that, so that we're getting really accurate input. But it turns out that's not true either. We'll touch on a little bit of this later, but our brains actually process all of our sensory information quite a lot before we're aware of it. And in doing so, often what we think we see and experience isn't quite the whole picture, because our brains like to take a lot of shortcuts in our perception. And then you might also see a lot of the time something about people being right-brained or left-brained, like right-brained people are creative and left-brained people are more like analytical and mathematic. But that's not true either. We typically use both sides of our brain pretty evenly. Although there are some functions that only occur on one side of the brain or the other, they communicate with each other really well. So that one doesn't quite hold up either. Hopefully I'll be able to demonstrate why that is. Okay, so to start with, we want to look at how we break down the anatomy of the brain into subcategories. And there are 4 main brain regions. And then there's some related structures, both surrounding the brain and inside of the brain, that kind of contribute to the overall anatomy. So we'll work our way through from the cerebral hemispheres through the diencephalon and the brainstem and the cerebellum. But we're going to start by talking about some of these related structures and their protective functions in keeping the brain safe. So just to give you an overview of what we're going to be looking at. So the cerebral cortex, cerebrum is up here. And then the diencephalon is in here. Ooh, excuse me. That was weird. The brainstem is here, and the cerebellum is here. So nervous tissue is quite delicate and easily damaged. So in order to keep the brain and the spinal cord from being damaged by even regular activity or pathogen invasion or anything like that, we have multiple layers of barriers between the central nervous system and the external environment. So we not only protect the brain and spinal cord from physical trauma, but we also protect it from chemical changes. Because as we talked about with action potentials, we really want a constant chemical environment so that the electrical activity isn't impaired. So the layers kind of go like this. We have the bones of the skull, creating that cranial cavity. And then we have the meninges, which are layers of connective tissue that we'll talk about next. And then the brain floats in the cerebrospinal fluid. And then there are protections keeping the chemical, keeping certain chemicals in the blood out as well, which we call the blood-brain barrier. Give me 1 second. Apparently I have to cough. All right, well, that's what I get for taking a walk right before I give a lecture. All right, so let's start with the meninges. So the meninges are actually 3 layers of connective tissue surrounding the central nervous system, so both the brain and the spinal cord. And so they offer a layer of physical protection in sort of a different way from the skull and the vertebrae, and they also kind of house all the blood vessels that are running into and out of the central nervous system, as well as all the nerves running into and out of the central nervous system. They also basically form the container within which the cerebrospinal fluid is located. And they partially separate different parts of the brain because there are some partitions of the meninges that kind of go down in between different areas. So the 3 layers are all named mother. So mater means mother. I don't remember in what language. And so the outermost layer is called the dura mater, which is tough mother. The second layer is the arachnoid mater, which is spider mother. And then the third layer is pia mater, which means gentle or delicate mother. So the dura is the outermost layer. And so it's going to be this dense, irregular, fibrous connective tissue. And around the brain, there are 2 layers to it. So the periosteal layer is actually connected to or attached to the inside of the skull. So it functionally is the periosteum. And then the meningeal layer is the one that actually covers the brain and the spinal cord. So this is in the cranial cavity only. And then this one is all the way around both the brain and the spinal cord. So then just deep to the dura is the arachnoid mater. So basically the connective tissue in here forms sort of a lacework, and it looks kind of like a spider web, which is where they kind of derive the name from. So there's a fluid-filled space separating the arachnoid from the dura, so that's the subdural space. And then there's a fluid-filled space, so CSF-filled, underneath the arachnoid between the pia and the arachnoid. So that's called the subarachnoid space. There's a lot of big blood vessels in there as well, in both of these areas. And there are these things called arachnoid granulations that we'll look at later that actually absorb the cerebrospinal fluid after it circulates around the brain and spinal cord. So we'll look at that part later. And then last is the pia. So the pia is just a very thin, delicate layer of connective tissue that follows all of the external contours of the brain and the spinal cord. And so every single little indentation of the external surface is followed with pia. And then as blood vessels enter the brain and the spinal cord, the pia actually follows them and kind of helps form a connective tissue sheath. So it looks like this. So here's bones of the skull here. And so we have the periosteal dura up here. Now it's fused to the meningeal, except when the meningeal goes down and divides or forms partitions. So then there's blood vessels, especially large veins, running in the dura. And then we have this subdural space that separates the dura from the arachnoid. And then the arachnoid is here. Hold on, let me switch colors. So then the arachnoid is here and has these fibers that extend down to connect it to the pia, which follows the brain contours. So this subarachnoid space is where the CSF runs. And you can see we have blood vessels coming through these things. And then this guy is an arachnoid granulation. So this is where the CSF gets absorbed. So it goes from here into the blood vessels in here to get absorbed into the blood. And then this guy is one of the partitions of meninges within the longitudinal fissure, which separates the right and left hemispheres of the cerebral cortex, which we'll look at in just a little bit. So speaking of cerebrospinal fluid, cerebrospinal fluid is basically just a filtrate of plasma, which is the liquid portion of blood. And as such, it's mostly made up of water and ions with a few proteins in it. So the main function of the CSF is literally to float the brain and the spinal cord so that it's not resting on its own weight, because the weight of the brain would damage the undersides of the brain tissue. So it floats the brain and the spinal cord. It also, because it's surrounding them in fluid, it protects it from damage. So if you've ever come to a really abrupt stop, you know, and like flung your head forward and stuff, the CSF is what keeps your brain from slamming into your skull and squishing. Okay, very important. And then we know that it has other roles as well, though we're still learning more about exactly how they work. But we think that it contributes to providing nourishment to brain structures, so carrying things like oxygen and glucose, and might also carry hormone signals, circulate them around the blood, around the brain. So CSF is both produced and reabsorbed in the skull. Sorry about that. So it's formed by these clusters of tiny blood vessels or capillaries called the choroid plexuses. And it's aided by the ependymal cells. So remember, the ependymal cells are glial cells, and their job is to create the CSF. So they basically control what exits these blood vessels and enters into the areas where the CSF are, and then those cilia on the cells circulated around. When they get back around to surrounding the skull or surrounding the brain, those arachnoid granulations absorb the CSF by allowing them a pathway into the blood that's located in the dural sinuses, which are the really big blood vessels that are in between the layers of the dura. So the whole pathway is, the formation actually occurs inside the brain. So there is a series of 4 ventricles. Somehow that doesn't make it on the slides. I don't know what happens. So there's 4 ventricles, and right in here, there's a 3rd ventricle. There's 2 lateral ventricles that basically sit on either side. And so all the CSF gets produced in there. That's where the choroid plexuses are. So they all get produced here, and then they circulate out. Here's the 4th ventricle. And then some of it goes all the way down the middle of the spinal canal, the spinal cord, and then some of it comes out and circulates outside of that and outside of the whole brain. And so it circulates, it goes all the way down, comes back up, and when they get back up here, that's when it all gets reabsorbed. So generally speaking, you're always making the same amount of CSF as you're reabsorbing, so that your volume is constant and your pressure is constant in that whole system. And so not only does the CSF surround the brain and spinal cord, there's internal areas of fluid as well. So here's one where we're highlighting the ventricles. So the 3rd ventricle is here. There's a left and a right lateral. So these guys sit on either side of the 3rd. And then the 3rd is connected to the 4th here. The 4th is connected to the central canal, which is the internal portion that holds CSF in the spinal cord. And then the rest of it is all external and going around both of them. So then our last type of protection is in controlling the chemical environment around the neurons. And we do that with the blood-brain barrier. So the blood-brain barrier is essentially extra protection on the blood vessels that supply neurons and glial cells with their nutrition. So capillaries are the smallest blood vessels. They're always the ones actually supplying tissues with oxygen and nutrients. And they are made of a single layer of simple squamous epithelium that we call endothelium. And normally these are fairly leaky, but in the brain, this capillary endothelium has a lot of tight junctions so that they're not very leaky. So tissue fluid can't really get in between these cells. It has to go through, which means there's a lot more control over what makes it out. The basement membrane around the capillaries are also fairly thick, so we just call it a basal lamina. And then the thing that controls this whole process is the 3rd layer, which are the processes of the astrocytes. So remember that the astrocytes are the glial cells that are named because they look like stars. They have a lot of processes, and some of those processes sit on the blood vessels. They thicken the basement membrane and encourage the capillaries to make more tight junctions. And then they ultimately control what is able to come out of the blood and into the extracellular fluid around the neurons. So this is just a diagram of that. So single layer of simple squamous here with tight junctions, and then a really thick basement membrane, and then the astrocyte processes. So because there's control, but things are still able to move, just like a plasma membrane, the blood-brain barrier is selectively permeable. And just like the plasma membrane, some things are still able to move back and forth without much trouble. So anything that can cross a plasma membrane is able to easily move across the blood-brain barrier. It can't be stopped. So anything that is like small and non-polar, like oxygen and carbon dioxide, or is fat soluble, So fatty acids and any other fat-soluble molecules can make it through because they can just cross freely and diffuse through those plasma membranes. The blood-brain barrier also doesn't try to stop glucose, amino acids, or most electrolytes. It wants those in. So they all have a lot of channels to make them move freely in. It prevents metabolic waste products, larger proteins, and some toxins from entering. And potassium is actually actively pumped out because we want the extracellular fluid around the neurons to have no potassium in it. And that's because it's going to make action potentials far more effective because we're going to have more of a gradient for when the potassium is going to flood out of the cell to repolarize after an action potential. Okay, so potassium isn't allowed out from the blood unless the neurons need more for some reason. Now there are a couple of places where the blood-brain barrier has gaps. Those areas are called circumventricular organs because they happen to be around the third and fourth ventricle. And circum is around and ventricle refers to those areas. And that's because a couple of parts of the brain have to monitor certain chemical conditions within the blood, and they need direct access. So they can't be behind a blood-brain barrier in order to be effective at their jobs. So a part of the brain called the hypothalamus, which we'll talk about later, has to monitor a lot of things in the blood, including pH and gas levels and all sorts of other things. And then we have something called the vomiting center in our brainstem that tells us whether or not we've potentially eaten something that we need to vomit back up again. So they both need to directly monitor the blood, and therefore there are little areas of vulnerability where things in the blood can actually get into the central nervous system because of these gaps. But like I said, they are necessary, so it's a trade-off. All right, any question about our protection before we get into our brain anatomy? All right, so we're going to start with the cerebral hemispheres, which are the superior portions of the brain. They're paired, so there's a right and a left side. And each side is divided into 5 lobes. The way we distinguish lobes is that there are deeper indentations in between them. Indentations in the brain are called sulci. And the lobes are organized so that there's an outer layer of gray matter, which we call the cortex, and then white matter underneath that, and then deeper areas of gray matter called the basal nuclei. Now the surface area of each lobe is increased by these gyri and sulci. So gyri are little up things and sulci are little down things. And so this is able to hold a lot more neurons than something that was just smooth and straight across. So that allows us to have a lot more neurons in there. Okay, so the gyrus goes up and the sulcus goes down. And when we have a deeper sulcus, then we're separating the cortex into different lobes. So here's the cerebral cortex. It's most of the external thing that we can see, okay? This longitudinal fissure divides them into right and left halves, so there's no nervous tissue touching here. There's some deeper in that we'll talk about in a second. So when we look at it with some colors to help us see, we can see that on both sides, we have a frontal lobe, a parietal lobe, an occipital lobe, and a temporal lobe. Now these happen to correspond to the skull bones that sit in those same areas, so that's handy. And there's a fifth lobe called the insula that we can't see from outside because it's deep. So insula is like insulated, right? Like hidden. And so that one is here. So if you look at it, basically there's this really deep sulcus that we can't see very easily, and the insula is right there. So if we go back, it's basically right here. And it kind of starts right there. All right, so 5 lobes on each side. All right, now the cerebral cortex is in charge of most of the higher brain functions, kind of what we think about when we want to talk about what brains do. What we think about is thinking. So this is where our conscious mind is. So we have an awareness of self and of our sensory input. This is where our ability to communicate, our ability to have memories, our ability to understand things, all those processes happen in our cerebral cortex. And it's also where we make decisions about movements. Some of these functions are located in very specific locations, and some of them are scattered throughout the cortex. So we'll just touch on that. We're not going to get into it in very much detail. So this is sort of just an overview showing you that a lot of the function locations are separated out by lobe. So for example, this is the part of the frontal cortex called the prefrontal cortex. And this is kind of where we have like planning and problem solving. We have hearing and things like facial recognition in the temporal lobe. We perceive most of our sensations using our parietal lobe, and most of our visual processing occurs in our occipital lobe. And then there are various spots for like language related things and other stuff in there. Now, connecting all of these things is the white matter of the cerebrum. So there are axons connecting all different parts of the brain to each other. So no matter what one part is doing, it's in communication with the other parts that support it. So everything is coordinated and based on a lot of crosstalk. So the types of white matter tracts, which are groups of axons, I'm pretty sure we talked about that, but I don't know. So groups of axons in the central nervous system are based on who they're communicating with. So association fibers connect nuclei. So remember, nuclei are groups of neuron cell bodies, also called soma, within a hemisphere. So they might be connecting the temporal lobe to the occipital lobe, or probably the frontal lobe to the parietal lobe, or something like that. But they're within the same cerebral hemisphere. So they're going to be involved in connecting up different types of input for coordinating like different senses and stuff. Commissural fibers are the ones that connect the left and right hemisphere together. The majority of commissural fibers are in a structure called the corpus callosum. And then in front of and behind the corpus callosum are the anterior and posterior commissures. And then finally, we're going to have the cerebrum connected to other parts of the brain and nervous system. And so projection fibers are the axons that actually leave or enter the cerebrum. So we kind of map it like this. So a longitudinal fissure separating the right and left hemispheres here. So the commissural fibers run just below the longitudinal fissure. So you can see that even though it looks like the right and left sides are pretty separated, there is a connection there. And they run pretty far along the length of this as well when you look at it from that point of view. The association fibers, like I said, they stay on one side or the other. So they're not crossing midline and they're not going up and down anywhere. They're just connecting different parts of each hemisphere to each other. Yeah. And then finally, we have these projection fibers, which are literally projecting axons either into or back out of the cerebrum, okay? So that's all the white matter associated with the cerebral cortex, or the whole cerebrum, sorry. And then the other thing we can see on here that's kind of nice is that you can see, here's the lateral ventricles, and then the third ventricle is more down here. So you can kind of get a feel for that. One of these days I'll find a good three-dimensional picture. Okay, so that's the cortex, 5 lobes per side. And then the other gray matter in the cerebrum is in what are called subcortical nuclei. So this is deeper gray matter. So that means it's still neuron cell bodies. And it's actually deep within the cerebral white matter. So we call them basal nuclei. You'll also see them called subcortical nuclei. And the old term, like literally, if you do a Google search for basal nuclei, you'll get a bunch of stuff talking about the basal ganglia. For some reason, it used to be called ganglia. And then they were finally like, you know, we should use different terms for gray matter in the central nervous system versus gray matter in the peripheral nervous system. And that's when they reserved ganglia for gray matter in the peripheral nervous system and nuclei for gray matter in the central. So old terms die very long deaths. So that's still floating around. But either way, this one is the most descriptive because it says it's underneath the cortex. But this works too because if something is at the base, then we know that it's nice and deep. So it's at the base of the cerebrum. So again, longitudinal fissure, corpus callosum, little bit of lateral ventricles here. So you can see all of the cortical gray matter superficially. All right. So then this stuff is all basal nuclei. It's in a couple of different spots. We're not going to worry about all their names or anything like that. We're just going to know that it's basal nuclei. So the basal nuclei have important functions as well. So they get communication with the whole cerebral cortex. And while we're still trying to figure out exactly what the details of their role are, overall, it seems to be about essentially editing what you're thinking and feeling and doing. So it's about removing incorrect and inappropriate responses to cognition and emotion. And if you're curious about how that works, there's a nice little one in that Brain Made Simple website here. And there's also a good one up here on this website. And then the other guys are a little bit more complex. So it just depends on how much you need to know. But this is all you actually need to know for this class, okay? They're like little editors or something. And then this is just a diagram showing you a little bit of that. So this is just a name for part of the basal nuclei. So this is showing you how the cortex is talking to the basal nucleus, and then it's doing things that you don't need to know about. And then it's talking to the thalamus, which is a lower part of the brain, which then talks back to the cortex, and it's this whole thing. So that takes us to the next part, which can collectively be called the midbrain. So in different textbooks, the midbrain is sort of everything that isn't the cerebrum or the cerebellum. But other textbooks divide up this into the two things that I showed you before, which is the diencephalon and the brainstem. So that's the way I like to teach it, because they do have very different functions from each other. But if you're like reading your textbook, you'll see it all called the midbrain. And so I just want to make sure that that's clear. So we're going to start with diencephalon parts and functions, and then we'll move on to brainstem parts and functions. So the diencephalon basically sits just under the cerebrum, and there are 3 paired structures, and they all happen to surround the third ventricle. So we'll go through them one at a time: thalamus, hypothalamus, and epithalamus. Now, the reason why they all have similar names is because they named the thalamus for something. I don't remember what it is. And then the other 2 are just described based on their relative location to the thalamus. So hypo means under, epi means around. All right, so the thalamus sits here. It's 2 little globes squished together, connected right and left sides by just this little stalk that runs this way. And then the hypothalamus is here, and then the epithalamus is like here on either side. So the thalamus has a really important job in that it controls all the information going into or coming out of the cerebral cortex. So it has a ton of different nuclei with different functions that ensure that all the sensory information coming in from the peripheral nervous system gets sent to the right part of the cerebral cortex, and that other information from different parts of the brain gets sent to the right part of the cerebral cortex, and then it makes sure that motor information from the cerebral cortex gets sent to the right place. So it's this massive relay station that organizes information and then sends it on its way. So there's a lot of synapses in the thalamus, because in order to do that job, it's all about synapses. So this is kind of our little 3D model. You can see it's like 2 little oblong-y things smooshed together with a little central connector. Sits right there and kind of goes through the third ventricle. All right, so that's that. Very, very tight communication between the cerebrum and the thalamus. So then we go to the hypothalamus. And the hypothalamus is like a completely different set of jobs. So the hypothalamus is the visceral control center. Its job is basically to do most of the stuff involved in maintaining homeostasis for all sorts of different things. It has a really strong connection to the endocrine system, which we'll talk about next week, because it's connected to the pituitary gland. So this little guy here, which makes a bunch of hormones. It also does a lot of stuff for the unconscious parts of our nervous system, which we'll touch on a little bit more when we get to the autonomic nervous system, because autonomic and visceral are basically interchangeable terms, okay? So very different job from the thalamus, but also very, very important. All right, So some examples. So for example, the hypothalamus will initiate physical responses to emotions. So if you're really excited about something and your heart rate goes up and you start breathing faster, it's because of your hypothalamus. If you get really scared and you start sweating and shaking or something, it's because of your hypothalamus. If you're really angry and you turn red and you also start sweating, because sweating happens a lot, that's your hypothalamus. It's also where our thermoregulatory center is, so it regulates our body temperature and controls the effectors that are related to that. It's the seat of a lot of things related to our appetite, so it controls food intake. It also monitors how hydrated we are and will make us thirsty when we need more water. It regulates our sleep-wake cycles, and through its connection to the pituitary gland, it actually controls endocrine function, which we'll talk about, as I said, next week. So kind of a big deal. The epithalamus is not nearly as exciting as the other 2. It's basically just a couple of little nuclei that have a variety of functions. We're just going to touch on one of them, which is the pineal gland. The pineal gland secretes melatonin when it's dark outside. Melatonin is a hormone that is involved in us having a circadian rhythm. So a cycle of different things over a 24-hour period of time, like when we're supposed to be sleepy and when we're supposed to be hungry and stuff like that. Okay, so when it's dark, we make melatonin. When it's not dark, we don't make melatonin. And we'll talk about it more when we get to the endocrine system. Okay, so this is the guy that gets really screwed up when you travel and you go to different time zones. So pineal gland's here, and the rest of the epithalamus is here, and we're not going to worry about what it does or what it is. All right, so that takes us to the brain stem, and it's called the brainstem because it's the most inferior portion of the brain. And there's 3 regions to it: midbrain, pons, and medulla oblongata. Now, overall, the functions of the brainstem are automatic behaviors necessary for survival. So, for example, breathing. And it's also the location of most of the nuclei associated with cranial nerves. And cranial nerves are the ones that control our face and parts of our neck. We'll talk about them later. So the brainstem is this stuff here. And when the brainstem ends, the spinal cord begins, and it's a very seamless process because it's really all the same structure. It all formed together. All right, so the midbrain has a lot of structures related to reflexes. So for example, it receives input from our eyes and our ears and lets us do things like keep our head still or keep our eyes focused on something even though our head is moving, which usually we're not aware that we're doing. It also does things like help us turn our head in response to a sound so that it knows exactly which direction it was coming from and it turns our head so we can look at it. Those are reflexive actions that your midbrain is in charge of. There are some things in here that help regulate our cardiovascular and respiratory systems, although most of that is done by other brainstem structures. And as I said, this is going to be true of all 3 of them. Nuclei for certain cranial nerves are located in the midbrain. There's not much to look at here. Should really just put the previous picture up because it's just kind of here. So like the hypothalamus is here, the thalamus is sitting on either side here, the pituitary and epithalamus is here. So the midbrain and the beginning of the brainstem is just after the diencephalon. All right, so the next up is the pons. Pons means bridge, and it was named that because so many axons, so many tracts run between the spinal cord and other brain centers via the pons. There are lots of nuclei in here for important things like breathing. It's part of something called the Reticular Formation, which is all about regulating our level of consciousness. I think I have slides on later. And then again, nuclei for a couple of the cranial nerves. Some of these connections between the spinal cord and other brain centers include a lot of connections to the cerebellum, which is the next part of the brain that we're going to talk about. And that's about basically balance and coordination. So we'll talk about that in a bit. So the pons is kind of the bulgy bit of the brainstem, makes it the easiest part to identify because it sticks out a lot like that. And that takes us to the medulla oblongata. So the medulla is literally like the most inferior part of the brain. So it blends with the superior spinal cord. So there's a big hole in the base of your skull called the foramen magnum. And depending on which way you look at it, either the spinal cord enters the foramen magnum or the spinal cord exits it. Doesn't really matter. It works both ways. So on one side of that hole, you have medulla oblongata, and then on the other side of the hole, you have spinal cord. okay? That's kind of how it works. So the medulla does a lot of the same things as the pons. It relays information between the spinal cord and the cerebellum and the cerebrum. It has nuclei for cranial nerves, and it has automatic function nuclei for things like the cardiovascular and the respiratory system. So you can see there's a theme here. So the brainstem as a whole is working together to regulate these functions. So here's the medulla, and then basically right here's the spinal cord. So, yeah, so a lot of homeostatic regulation comes with these automatic and autonomic reflexes, and that includes other reflexive activities as well. So things like sneezing and coughing are also regulated by the medulla. So there's really no one part of the brain that's more or less important than the others. They all do something different that's still super important. Which takes us to the last part, which is the cerebellum. It's also called the hindbrain because it's the most posterior portion of the brain. And the cerebellum is all about coordination. So the cerebellum coordinates motor output so that movements are smooth and timed and agile. And when the cerebellum doesn't work, well, bad things happen. I'll show you. One second. I have to go out of this, and I'll do another one with the internet. Oh no, no sound, no sound. Okay, sorry. Just establish that I have to screen share before I go to full screen for this as well. Doesn't work. All right, so this is a cat whose cerebellum doesn't quite work right. I'm sure that most of you have seen a cat walking before, and if not, you've probably seen a dog or some other animal. So you might notice that this cat does not walk normally. It kind of walks like, can't quite seem to put its feet in the right place, and that's making it very uncoordinated. And yet it's still willing to try normal cat things like climbing into a bathtub and playing with stuff. Even though its legs go all over the place, it still manages to get down the hallway. So that's because it knows where all of its parts are and it knows how to move them, but its cerebellum isn't very good at fine-tuning those movements. So the cerebellum is basically constantly monitoring what your body is doing and where it's located in space. And when your muscles try to do too much, which is usually the case, or too little, it corrects that. It says, no, we need this much muscle effort, not what you're trying to do. No, you need to move your limb this way and not where you just tried to go. And without that, or without as much of it, you end up with this. And if you lose too much cerebellar function, then in cats at least, we get an animal that can't walk because it literally cannot get started. And yet we can see that with some function, this cat can do all normal activities. It just might fall over every now and then and look kind of goofy while it's doing it. And yet the super odd thing is that there was recently, let me go back to the lecture. There was recently a woman who in her 20s, it was discovered that she doesn't have a cerebellum. There's just cerebrospinal fluid where her cerebellum should be. And yet she walks and talks and I think moves mostly normally. It wasn't quite clear exactly what had gone wrong to take her to the point where they did imaging and found out she was missing an entire portion of her brain, but clearly she had compensated for the lack of cerebellum. So there's that flip side to it too, because brains can adapt if things happen, especially if things happen early enough along. So here's the cerebellum. Now the name cerebellum just means little cerebrum, so it's a little brain, because the cerebellum looks like a miniature version of the cerebrum. So there's right and left hemispheres. The hemispheres are divided up into lobes by deeper fissures. There's gray matter cortex and deeper white matter. The white matter is arranged in a way that it kind of looks like a tree, so they named it the arbor vitae. So here's the picture in your book. I think I put, yeah. This one's a little bit more close up so you can kind of see it better. So this is through one of the hemispheres. So we can see 3 lobes, right? And you can kind of see how this looks like a tree, the white matter deep in there. And then the cortex has kind of the same sulci and gyri effect as the cerebrum. So we're packing a lot of neurons into this relatively small brain structure because the cerebellum has to do so much. Anytime you're moving, cerebellum's working really hard. And you can see the pons is right here, so connections to the pons are pretty easy. So basically how the cerebellum does this job is that when your cerebrum decides that it's going to do something, so when it makes a decision to initiate muscle contractions, cerebellum gets notified. It's like, okay, you're going to do this, right? You're going to pick up this cup. So as you're doing it, the cerebellum is getting this moment-to-moment feedback about where your body currently is, and as that changes, where it's going. So you reach for your cup and you pick up your cup, and your cerebellum is getting that input the whole time. So your cerebellum is going to do these calculations, these moment-to-moment instantaneous calculations about how much muscle contraction you need, which muscles have to contract, how much, to not only keep yourself in whatever posture that you think you should be in at that point in time, as well as smoothly do this thing so that you can reach up and grab a glass of water and drink it and not, you know, miss the glass and knock it over, or bring it back over here, but you're shaking so much that the water is splashing, or try to drink it but miss your face. All sorts of things can happen because it turns out that the cerebrum all by itself isn't very good at controlling your muscles in an adequate fashion. So we have the cerebellum too. In order to actually make those corrections, the cerebellum sends its plans back to the motor cortex as well as the brainstem, and then they carry out the corrections. And so this is happening constantly whenever you're moving. And even when you're not moving, if you're using your muscles, it's still happening. So it's involved in helping you maintain your posture, whether you're standing or sitting or whatever you're doing. All right, so that is all of the brain as far as the anatomy. There's a couple of things that are good to point out too, which is that some of the brain's functions occur in scattered structures throughout the brain. And so while we don't necessarily have this single area that does it, we have what we call these functional brain systems. So there are networks that work over large areas of the brain to do a specific thing. And we're going to talk about the 2 main ones very briefly because they're fairly important, just like all the other things we've talked about. So the first one's the limbic system. And the limbic system is a variety of structures that sit in and surround the upper part of the brainstem, and they function as our emotional brain. So any sort of emotional responses that we have are going to be because of your limbic system. Our ability to express emotions is also thanks to our limbic system, and how we process our emotions is also going to involve the limbic system. The limbic system is often triggered by odors and memories because there's a lot of cross connection between the parts of our brain that process smells and the parts of our brain that process memories. And not surprisingly, there are connections between the limbic system and a lot of different brain regions so that we can have appropriate emotional responses to a variety of environmental inputs. So that, you know, when you, I don't know, I got nothing right now. Just think about anything that happens that you have an emotional response to, and that's because your limbic system is tied in, okay? It's also connected to our prefrontal lobe of the frontal lobe, which, as I mentioned before, is involved in like our higher level thinking and processing. It's kind of our rational brain. Well, it's not that great at it. And so our emotional brain and our rational brain are tied pretty closely together, which is why we can do things like think about our emotions and have emotions about our thoughts and vice versa. So all of these different colored things are the various structures of the limbic system, and they all have names and they all do stuff, and we're not going to worry about that. The other functional system is the reticular formation. So the reticular formation is throughout our brain stem. So literally throughout the midbrain, pons, and medulla. There are various nuclei, and they govern the arousal of the brain. So our state of consciousness. And the reticular formation has input and output. And the output part is called the reticular activating system. And that's literally what keeps your cerebral cortex awake and able to respond to stimuli, so excitable. And when it fails, you go to sleep, basically. So it does that by filtering sensory input, and it does that by stopping your sleep centers from putting you to sleep. It also has some motor functions involved in your like internal organs and some of your limb movements. So coarse limb movements mean like when you're using like your big strong muscles instead of fine movements where it's like you're using your fingers and stuff. So this is kind of a diagram of the reticular formation. So the nuclei are all here, and then there are connections between the reticular formation and the hypothalamus for that visceral control, and then to the thalamus so that there's control out to the cerebral cortex so that it's able to keep us awake when we're supposed to be awake and then let us go to sleep when we're supposed to go to sleep. Does anybody have any questions about the brain? All right, so we're going to move on to the spinal cord and hopefully touch on, or at least start with our nerves. So remember that the central nervous system is the brain and the spinal cord. So we did this, and now we're going to do this. So the spinal cord is a literal extension of the brain. So it all forms together as a single nervous tissue structure. And as I said, it starts as the brain exits the skull at the foramen magnum, which is literally the large hole at the base of your skull. In adults, the spinal cord only extends down to the very beginning of the lumbar vertebrae. And that's because it doesn't grow as much as our bones grow. So it's much farther down when we're born, and then as we grow overall, it slowly kind of recedes up higher and higher. Now the nerves run all the way out, and we'll talk about that later. So the main jobs of the spinal cord are as the pathway between our sensory input and the brain, and then the motor output from the brain back to our effectors, so our skeletal muscle, smooth muscle, and our glands and stuff. There's also processing that occurs in the spinal cord. So even though the brain does most of the integrative function of the nervous system, there is neural integration at the spinal level. And a lot of that integration is with reflexes. So reflexes are unconscious actions that happen usually for protective purposes. We'll touch on a couple of them. And the reason why a lot of them happen at the level of the spinal cord is because it's faster. If a reflex has to go, say, from the base of your spinal cord, because it has to do with your leg, all the way up to your brain and then all the way back down again, we've lost precious seconds. Whereas if it can just get processed in the spinal cord, then we can have that appropriate reflexive response. There are also reflexes that help us do things like walk automatically. So we have these pattern generator circuits in our spine. So like when you make the decision to start walking, sure, that comes from your brain. But when you're just like going for a walk and you don't think about where you're walking or how you're walking, you're just walking, now your brain has let your spinal cord take over because it's this rhythmic pattern of muscle contractions and relaxations. And so your spinal cord is able to do that. The anatomy of the spinal cord is both similar to and different from the brain. So just like the brain, the spinal cord is protected by a layer of muscle, the 3 layers of meninges, and it is both surrounded by cerebrospinal fluid and has cerebrospinal fluid running down the middle. Some differences. The dura mater is just the meningeal layer, so there's no periosteal layer stuck to the inside of the bone. Instead, there's actually an epidural space separating the inside of the vertebral bones from the dura, and it is actually filled with fat, which is an additional protective layer, actually. The arachnoid mater is stuck to the inside of the dura instead of there being a space in between them. And then the pia actually has little bits of tissue that stick out and stick to the arachnoid mater, called denticulate ligaments along the whole length of the spinal cord, which helps the spinal cord stay anchored in the appropriate position so that it's not falling and trying to pull the brain out, because that would be really bad. We divide the spinal cord into sections that correspond to the vertebral sections. So we have cervical vertebrae, so we have cervical spinal cord. We have thoracic vertebrae and thoracic spinal cord. We have lumbar vertebrae and lumbar spinal cord segments. And then even though the sacrum is a single fused bone, it formed as multiple vertebrae. And so we have multiple sacral spinal cord segments. And then we have a single little coccygeal segment as well. After the coccygeal segment, or at the level of the coccygeal segment, the spinal cord terminates in the cona medullaris. So basically it literally just tapers down to an end. And then the filum terminale, which is anchored to the end of the meninges, so that not only are there ligaments holding the spinal cord throughout the length of the vertebral canal, there's also something holding it down so that it doesn't pop up either, because that probably wouldn't be good either. Below the level of the spinal cord within the vertebral canal, there's a subarachnoid space called the lumbar cistern, which should hold CSF. So when we look at this on cross section, we see the bone of the vertebra here, and then we have the epidural fat, which fills in the space in there that isn't filled with spinal cord stuff. Then we have meninges, so this is fused dura and arachnoid, and then we have the subarachnoid space filled with CSF. And just like in the brain, the pia is adhered to the whole outer surface of this. What am I forgetting? Oh, the denticulate ligaments never get pictured, but they basically do this. And we're going to talk about these nerves coming off in a little bit. So this is more of a longitudinal view. And here we can see here are the denticulate ligaments. So they're anchoring the pia to the overlying arachnoid. And that, like I said, helps everything stay in the right level. So most of the anatomy that we appreciate in the spinal cord is through a cross section, which is like the first picture I just showed you. And there's a butterfly on here because we have central gray matter, and it actually kind of looks like a butterfly. And then we have a surrounding white matter. So that's different from what we have in the cerebral cortex, right? So the majority of gray matter in the brain is in this thin external layer. And then the spinal cord is set up like the rest of the gray matter in the brain. So all those basal nuclei where it's surrounded by white matter. There are a fissure and a sulcus dividing the left from the right, partially, so not unlike in the brain. And then there is a canal in the middle called the central canal that CSF runs down along the entire spinal cord. The gray matter is organized into horns, and the white matter is organized into columns, and I will show you that on the diagram. So, cross section. So the right and left are divided by this posterior median sulcus and this anterior median fissure. The central canal is right here in the middle of the bit of gray matter that actually crosses from right to left. And then we have a posterior horn, a lateral horn, and an anterior horn in the gray matter. We have this little gray commissure, because commissure is what we're going to call anything that crosses from right to left, or, you know, left to right, either way. And then we have columns of white matter. So white matter posterior, white column, and then lateral white column, and then anterior white column. And then we have this these white commissures as well, anterior and posterior. Technically, the gray matter is anterior and posterior as well because of the central canal in between, but I'm not too worried about the commissures. Really just need you guys to know about the columns and the horns. So one of the reasons why it matters that we know about the horns and the columns is that they are organized based on the type of information that is in there. So the dorsal horns of gray matter receive sensory information from the body. The ventral horns send out motor information from higher levels of the central nervous system. And then the lateral or intermediate horns contain autonomic neurons and do something else that we'll talk about later. Sometimes it's motor, sometimes it's sensory. There's also organization in the white matter. So the columns are organized by ascending tracts, which are carrying sensory information to the brain. Descending tracts, which are carrying sensory information from the brain to effectors. And then transverse tracts are what we call the commissural fibers. So the ones going from right to left, left to right. Now, the white matter isn't quite as neatly organized between sensory and motor. So you can see this is showing us dorsal columns with sensory, lateral columns with sensory, ventral columns with sensory, and then we've got motor here and here as well. Okay, so with the gray matter, this is very much sensory and motor, but it's not quite like that with the white matter. And you do not need to know any of these specific ones, okay? All right, any questions? That's spinal cord. We talk about the brain forever, and then we get to spinal cord, and it's very fast. Okay, so we should have time to talk about the anatomy of nerves, and then we'll fit the rest of it in at some point. So the peripheral nervous system. is mostly the axons of neurons whose cell bodies are in the central nervous system, which I think is very interesting. And so most of it is going to be nerves, because nerves are bundles of axons. And we organize those into either cranial nerves coming from the brain or spinal nerves coming from the spine. And then remember that there are neuron cell bodies in the peripheral nervous system. Those are called ganglia, and we'll get to where those are later. Or now. Either way. Sorry, I did a lot of editing, and I can't always remember where every single slide is. So, ganglia, 2 locations. The first one is the dorsal root ganglia. The dorsal root ganglia are sensory neuron cell bodies. So these are those unipolar neurons that we talked about before. So they have these dendrites at the sensory receptor area, and then the axon is really, really long, and most of it is actually not technically after the cell body. So then the cell body's here, and the spinal cord is right here. So they're right on either side of the spinal cord, and so we call them the dorsal root ganglia, or DRG. Now, the other gray matter in the peripheral nervous system is all autonomic. So it all has to do with the visceral nervous system, and it's pretty much all motor neurons. So these are motor neurons whose cell bodies are in the peripheral nervous system, and then they're sending their axons to visceral effectors. So that's smooth muscle and glands and cardiac muscle. These guys are also located on either side of the spinal cord, and then some of them are located near some of the internal organs. I'll show you some pictures of that when we get to the autonomic nervous system. So the dorsal root ganglia, so here's our spinal cord again. So gray matter, white matter. Now, the connection between spinal nerves and the spinal cord is 2 roots. We'll talk about it again later. So the dorsal root carries all the sensory information from the sensory neurons, and the neuron cell bodies are here in the dorsal root ganglion, because it's literally a ganglion in the dorsal root of the spinal nerve. And then the ventral root carries all motor information, including some of the motor information for the visceral nervous system. And so the visceral nervous system ones have a ganglion that's kind of just off of the spinal nerve as well. So they're both right next to the spinal cord, just outside of the vertebrae, or, well, this one's, it's complicated anyway. And then there's some that are elsewhere, but we'll worry about those later. This is where almost all of the gray matter in the peripheral nervous system is. You'll notice how close it is to the central nervous system. Okay, so that's gray matter. Now we're looking at white matter. So white matter in the peripheral nervous system is in nerves, where nerves are literally bundles of axons running parallel to each other to different locations. Now, axons are actually organized very similarly to muscles. So we're going to have 3 layers of connective tissue surrounding the entire thing. We're going to divide up the axons into bundles called fascicles, and the entire thing is going to have an epineurium, so a dense, irregular connective tissue sheath surrounding the whole thing and protecting it. The fascicles are going to be surrounded by a perineurium. We're going to have blood vessels running through between fascicles and then into the fascicles. And then each axon is going to have an endoneurium surrounding it, as well as Schwann cells if it's myelinated. Each nerve has a very rich blood supply because all these guys have to get a lot of oxygen and stuff. So if we look at this in a diagram, epineurium surrounding the whole thing, fascicles or bundles of axons, so perineurium surrounding these guys with blood vessels running through, And then if we look at a single fascicle, we have perineurium and endoneurium around each one. All right. So we're using the same words, it's just that now instead of mesium, we have neurium instead. So instead of muscle, we're using nerve. Hopefully that makes it fairly easy to remember. All right, any questions? Where are we at? We can do it. All right, so the next thing we want to talk about is the general functions of the specific nerves. So we'll start with cranial nerves, and there are 12 pairs of cranial nerves, and they're coming out of or going into the brain. Most of them are coming out of or going into the brain stem, and we see pretty much all of them on the inferior aspect of the brain, and they control all of the stuff on your face, head and neck mostly. There's some exceptions that are not cranial nerve. Now, the cranial nerves are named by both their structure or their function, and they're given a number. So they're numbered in Roman numerals from rostral to caudal. So with Roman numerals, that's a 1, that's a 2, 3, that's a 4, because this is a 5. So 4 is just 5 minus 1, 6, 7, 8, 9 is actually 10 minus 1, so this is 10, right, and 11, and, oh god, that was 11 and 12, okay? So they're all numbered as well, unfortunately. And when we say that they're rostral to caudal, so this is the anterior aspect, this is posterior. So for reference, your eyeballs are like right here, okay? So we label 1 through 12 this way as they're seen emerging from the base of the brain, okay? So we'll start with the first one. So cranial nerve 1 is called the olfactory nerve because it carries all of the sensory axons for smell. We'll talk a little bit more about smell when we get to the special senses. And it's real small, it's right there, and it's the first one. Cranial nerve 2 carries all of the information from your eyeballs, so it's your vision nerve. So all of the vision-related information that hits your eyeballs gets carried in your cranial nerve 2s, which are called the optic nerve. And the interesting thing about this one is that half of it crosses over, and we'll talk more about that when we get to vision. Why did I, oh, right, yeah. Now we happen to have 6 muscles inside of our bony cavities where our eyeballs are, and they let us move our eyes back and forth and up and down and whatever. Cross your eyes, whatever you can do with your eyes, roll them. So there's 6 of them, and they're basically just named by the angle that they come onto the eye. So 4 of them attach the eye very straight. We call them the rectus muscles, and we just name them by their relative location to the orb. And then the other 2 come in at an angle, so we call them oblique muscles. And one of them is up top, so it's the superior oblique, and the other one's on the bottom, so it's the inferior oblique, okay? And I mention this because some of the cranial nerves control these muscles. So the oculomotor is cranial nerve 3, and it controls 4 out of 6 of those eye muscles. So basically it controls the inferior oblique, and it controls the superior medial and inferior rectus, okay? And then just some other things, we're not gonna worry about it. The 4th one is the trochlear, and the trochlear supplies just the superior oblique muscle. And it's called the trochlear because the superior oblique runs through this little pulley thing that's attached to the bone, and trochlea means pulley. So it has to go through that to hit the eyeball at the right angle to then, you know, let you like look up and in. It's basically what happens when you use that muscle. Okay, so that's trochlear. So we only have one eye muscle left. But before we get to it, we have to hit the trigeminal nerve. So the trigeminal nerve is actually the physically largest cranial nerve. That's cranial nerve 5. And it's largest because it carries all of the sensation from your face. And your face feels a lot of stuff. So all that sensory information comes from your face in 3 different branches, which is where the word trigeminal comes from, because it's literally this giant nerve with 3 branches. Okay, you don't need to worry about the branches. And on top of that, it controls the muscles you use for chewing. So all those jaw muscles. Got one there, got one there, got a couple more in here. Okay, so that's trigeminal. The last eye muscle one is abducens. It's cranial nerve 6. So this is the lateral rectus muscle that it controls, which is actually this one that they've shown cut here. And the reason it's called the abducens is when you use your lateral rectus muscle, it makes your eyeball look laterally. So you can only do it with one at a time, but you're looking to the side, away from midline, which is how we describe abduction. It's movement away from midline. So they called it the abducens. Super clever. 7 is facial. So whereas the trigeminal nerve is all of the sensory information from the face into the brain, the facial is all of our motor control from the brain out to all our facial muscles, other than the ones we use for chewing. So we say it's the motor to the muscles of facial expression, and then it also controls most of the glands of our face too. So our tear glands, our nasal glands, the ones that make all the mucus, and then all of these guys are most of our salivary glands. And then it also carries information for taste, but just for the front of our tongue, the anterior 2/3. Why? I don't know. That's the way it works. So a lot of taste, but not all of the taste, goes by your facial nerve. And you don't need to know this, but there are 5 branches. It's also fairly large. 8. 8 is vestibulocochlear. And it's called that because it carries the sensory information from 2 special senses from your inner ear, your vestibular sense, which is balance and equilibrium, and then your ability to hear, which happens in this structure called a cochlea, which is how we get to vestibulocochlear. All right? Don't worry about that. I took that out. Nobody cares. Okay, so that's 8. Special senses. We'll talk more about them later. How have I never fixed this? Oh, sorry. This is 9, glossopharyngeal. Now, glossa refers to the tongue, and the pharynx is the muscular part of your throat, just behind your oral cavity. So this muscle is going to have control over your tongue and your throat. So the rest of the tongue's tasting abilities are sent back to the brain by this nerve. The last salivary gland, the parotid's actually the one back here, or I guess back there, is controlled here, as well as parts of our ability to swallow, because the pharynx has muscles that are involved in swallowing. So that's glossopharyngeal. It's kind of a weird catch-all for part of the throat, essentially. And that's going to take us to 10. 10 is the vagus nerve. Now, the trigeminal might be the largest, but the vagus is arguably the most important. And this is because it's the only nerve that controls anything outside of the head and the neck. So not only does it have motor control over parts of the pharynx as well as the larynx, so that's the rest of your throat for swallowing, so it works with your glossopharyngeal, but it also controls a lot of the autonomic output for your visceral organs. So your thoracic and abdominal organs. So we're talking lungs, heart, all your digestive organs, a lot of other things. Lots and lots of, oh, there's like kidney and spleen in here too. All of your soft organs basically have some amount of control by your vagus nerve. So that makes it super important. So here's a little diagram for what the vagus nerve does. So the swallowing component's up here, and then all of these connections to various organs farther out in the body. So it's the only one that does that, the only one that leaves the head and neck. So after the vagus, the other two are kind of, you know, less exciting. The vagus is also interesting because we think that certain conditions can be treated by electrically stimulating it. So that's information about that if you're curious. But 11 is the accessory nerve. And the accessory nerve actually controls the trapezius muscle and the sternocleidomastoid muscle, which are two major muscles of the neck area. The trapezius is actually a major muscle of your back, but that's besides the point. It connects to your head. So that's the one there. That's the one where when you build it up, you bulge here. And then your sternocleidomastoid runs along the front of your neck from your collarbone up to the back of your skull. So it's a major neck muscle. And it actually used to be called the spinal accessory or accessory spinal nerve because it looks more like it's coming off of the spinal cord rather than the brainstem. And it kind of is, but that's a whole other can of worms. Now that takes us to our last cranial nerve, which is 12. So 12 is the hypoglossal. Hypo means below, glossa again means tongue. This is where we finally have the ability to actually control our tongue. So the other ones were just about relaying taste information, but this one actually lets us control our tongue so that we can chew food, so that we can swallow it, and so that we can manipulate our tongue to make different sounds for speech. So the tongue is complicated, obviously. Oh, thought I had my brain picture there. So anyway, if you, nope, oh well. Any questions about the cranial nerves? So you guys just want to know their name, their number, and their general functions. Okay, so we will finish up the spinal nerve thing on Wednesday before we launch into the other stuff. It won't take that long, but that was a lot of information, so we'll stop it here. Anybody have any questions? Oh, chat. I finished grading exams. No, I'm sorry. I've been working on it, but I'm hoping to get them done by tomorrow, and so then you'll all have them back. And yeah, there's been a lot of grading, so a lot of grading, and then I had to make another exam too, and then I'll have to grade that after I grade yours. So I'm steadily working my way through them. But your exam is the next thing that I should be finishing. Any other questions? Next exam is Friday. So yeah, the current schedule is so that the next exam is Friday. So basically the goal is to finish all our new material by Monday, or at least almost all of it. And that way Wednesday can be review. So if you have any questions about any of this, and then Friday seemed like a good spacer day for the exam, but I am happy to bump it back if there's an issue. It's not set in stone or anything. Any other questions? All right, if that's it, then I'll see you all Wednesday, and we'll keep talking about the peripheral nervous system.